All right, welcome everyone to today's BTEP Coding Club event, an introduction to David for functional enrichment analysis. My name is Alex Emmons, and I'm a bioinformatics trainer with the NCI CCR Bioinformatics Training and Education Program. Today, we welcome guest speakers and developers of David, Weizong Chang and Brad Sherman of the Laboratory of Human Retrovirology and Immunoinformatics Lab. They are going to introduce David and show us how to use some of the key tools. So we're going to hold our questions until the end of their presentation, at which time you can either unmute yourself and ask your question directly, or you can put your question in the chat and myself and my BTEP colleagues can get those questions over to the speakers. Today's event is being recorded, and that recording will be made available 24 to 48 hours following the event in our BTEP video archive. And along with the link to the archive, I will also put the event webpage, which includes the slides and a few other files in the chat so that you can maybe test things out following the presentation. So with that being said, I'm going to go ahead and hand things over to our speakers and we'll get things started. So thank you for being here.
Thank you, Alex. Good morning, everyone. We are very glad to have the opportunity to introduce the David for functional enrichment analysis to our community. I'm the Weizong Chen, the head of bioinformatics section in the LHRI lab. And Brad Sherman is the senior bioinformatician in our section, and he is one of the major developers of the David. So today's tutorial includes two sections. I will give a very brief overview of the David, and Brad will give you the live demo on the David. And we also include the demo screenshot in this slides, so you can follow along after the tutorial. So David is the abbreviation of the database for annotation and visualization and integrated discovery. It is a bioinformatics resource consisting of the knowledge base and analytical tools. It aims to systematically extracting bioinformatical meaning from the larger gene and protein lists. So it can provide the investigator with the ability to gain in-depth understanding of the biological themes that are enriched in the genome scale studies, such as the microarray assay in the early age, and also RNA-seq, single RNA-seq, chip RNA-seq, currently used in the field. And we have new URL moved that is just recently obtained for the NIH. So it is called davidbioinformatics.nih.gov. So I also include in this slide. You can access later. The David first published in 2003. Since then, there's 11 major version update. All is linked because of the big update in the public available database in the NCBI and the UBI. So that is so we can have the David current for all the time. And since last major update in January 2021, we also implement the quarterly update. So now is to make sure all the knowledge base is current. David is very popular in the scientific field. Since 2003, there's 11 papers published in our section for the related to David. As of March 21st this year, we have more than 55,000 citations for this David paper. And David consists of the knowledge base and also 6 tools. And there's 2 utility tools, the gene batch viewer, gene ID conversion. There's 4 annotation tools, the functional annotation table, function annotation chart, functional classification, and functional annotation cluster. All these tools can be accessed through the web interface or the web API or the web service. And Brad will give you a demo of all these 3 types of access today. And if you have any question, you can ask at the end of the demo, and also you can contact us through the email. Thank you very much. Now I will hand over to Brad. So let me see how I stop sharing.
Thank you, everyone, for giving us this opportunity. Can you hear me okay?
Yeah, we can hear you.
Great.
So
and can you see my screen?
Yeah.
Okay. As Weizong talked about, there's a lot to David. I'm going to do my best to get through it. I know we're limited on time, and there are a lot of different pieces to David. Just to point out on the home page, or any page really in David, there are several links in this main menu that can provide different forms of information. Also, I would encourage you, if you have questions, to contact us. There's several ways that you can do that are listed here. So what we're going to do is we're going to start an analysis. Hopefully this goes smoothly and quickly. So we'll click on this start analysis, which brings us to the analysis wizard. And the first thing that David wants us to do is upload a gene list. So this is the most important step. What is a gene list that should be submitted to David? It's one from your experiment that you've already done statistical analysis on. So differentially expressed genes. It is not like other tools like the gene set enrichment analysis where you put everything in. It does expect that these genes are significantly associated with your experiment, and that's done ahead of time. There are several different ways to get a list into David. You could simply paste a list of gene or protein identifiers. So let me pick my couple of favorite gene symbols, interleukin-2, interleukin-6. So this would be the first step, to either paste a list. It can be comma delimited, new line, space, tab, anything like that. And hopefully David will be able to pull those apart into individual identifiers. Or we can choose to upload a file. And a file for this upload needs to be tab delimited. So here is an example of one. This is our demo list 2, which is one that I'm going to use for the demonstration purposes. It's actually from a microarray experiment that looked at the GP120 in HIV inducing cytokines, chemokines, kinases, and transcription factors. So again, tab delimited file. The first column is expected to be your identifier. In this case, it's an Affymetrix ID from that microarray. Any following columns are considered by David to be a value. It's just something that we can pull in. We don't use it currently. Hopefully in the future we can put this to use if this was something like fold change, but you can carry it along if you chose to include that. Okay. With the file upload, there is also a different type of file that we can upload, which is called a multi-list file. So if I wanted to upload several lists with one file at the same time, so for instance, if I can do it, something like this, where I have both our demo lists. So again, this is tab delimited. I have a header, which is going to become the list name, and then I have the identifiers, and again, these are Affymetrix IDs. It's really important that these are the same identifier type. So in that case, if I uploaded that file, after checking this, I would have 2 lists now uploaded in David. But let me start with these 2 gene symbols, because gene symbol is a special case in terms of uploading. The second step after either pasting or the file upload is we want to select our identifier type, and there's many that we can see here. One is official gene symbol, which I'm going to use. At the very bottom, there's this choice for not sure. And if I select that because I'm not sure which identifier I have, then David will try to help you with that by redirecting to the conversion tool, which I'll talk about a little bit later. So I'm going to select official gene symbol here. And as a special case for gene symbols, because they are ambiguous across species, one symbol can mean multiple things. I need to tell David what species this is. I can enter the taxonomy ID. I could type in the species name, but nonetheless, it should give me a list of choices. I'll choose Homo sapiens here. The next step is to select what list type this is. So in David, for the enrichment analysis, we have our initial differentially expressed gene list, but then we have to also have a background from which we selected that gene list. So it could be the background species. The demo list that I'm going to show is from a U95A Affymetrix microarray, so that should ideally be the background set. But nonetheless, if I'm just coming into David for the first time and I do not have any gene lists loaded, I have to upload a gene list first. And that's what this is, despite being small. I'll go ahead and submit that. That gets uploaded. And in this list manager tab, I'm shown some information about that list, a default name here, which I can change. I can remove this list. If I had multiple lists, I could combine this list. But let's go back to upload. And I'm going to simply use these links up here to upload my demo list. So it's the same exact list that's here that I could upload as the file. But I'm just going to click on it here. And then I'm going to talk a little more about this, this list manager here. So we have the upload tab that we went through. The list manager is going to show me what species are in my list. Ideally, you want to work on one species at a time because typically, again, your background is going to be chosen from one species, and that's important, very important for the enrichment analysis statistics. I can see here that initial list that I uploaded, and then my demo list, which is now the current list because I just uploaded it. But if I wanted to switch between them, I could do that by highlighting and selecting use. Okay. So now that my list is uploaded, the analysis wizard shows me a little information about what's going on. So currently, I have demo list 2 loaded as my current list, and my current background is Homo sapiens. So go into this background tab, it'll show me that. And in the same way, I could rename these or use these. There are several pre-built backgrounds here, some Affymetrix IDs, some Agilent arrays, sorry. Or I could actually go back to the upload tab and upload my own specific background. But for this demo, I'm just going to stick with Homo sapiens since it's the default. And so now that I have a list uploaded in David, I can start to use the tools in David to analyze my list and learn more about the enriched biological themes that are occurring. So the first and simplest tool is the gene name batch viewer. So I can get to it from this link. I can get to it from the show gene list link. I can get to it from the shortcut to David tools. So in David, there's several ways to get to these tools. I'll click here. And what this does is gives me an initial look at my gene list. So the fact that I have Affymetrix IDs, they're not always very informative. I don't know what 1005_AT is, but now I have a gene name. So I can tell now what kind of genes I have in here just simply by the name. So it's a quick look at what my list is. This value, again, is what was pulled in along with that file. Again, it just gets pulled through the analysis. We're not using it right now. And then there are a couple of other pieces of information here. There's something called related genes, which I'll show in a minute, and then the taxonomy, which is linked out to NCBI taxonomy. So each gene name is actually linked to a gene report. So if you wanted more information, quick information about a specific gene, you could click on that and get that information. Related genes is a little more advanced. So what this does is looks at the genes within my list and compares the annotation, the functional annotation that they have in common and that that are not uncommon. And using a statistic called a kappa statistic and that matrix, binary matrix, we can assign a similarity score. And that's what this is. So in the case of the gene I clicked on, it was this DUSP1 gene. The kappa score is 1 because it is compared to itself. It is a perfect score. If we look at this other one, which is DUSP4, which we can tell is very related, right? Just by the gene name alone, we can now see that, give it some kind of a similarity score here. So if I click on that, I can see a 2x2 table that outlines the numbers of annotations that these two genes have in common, the numbers that are uncommon, so for this one compared to this one, or here, or the numbers of annotations that I searched, in this case 352,000, that are not annotated to either one of these genes. So using that, we can create this kappa similarity score. So we can find genes within our list that are very similar to each other, or if we go to the options, we can actually look in our background list. So in this case, Homo sapiens, I can rerun this. Hopefully it will run efficiently. The two genes I saw before from my list are highlighted in yellow, and then these other genes are related genes in that background species. And I'll come back to this kappa statistic and related genes in some of the later tools. Okay, so if we go back to the wizard, that covers gene name batch viewer. I'm going to then move to this group of functional annotation tools. So I'll click on that. And what I see is a summary of all the annotation in David that is associated with genes in my list. And they are organized into main groups. So for instance, disease, I have DisGenet, Genetic Association Disease Database, OMIM, UniProt keyword disease. Some things that I can see are percentages here, which is showing how many of my genes in my gene list are associated with at least one term in this database. So that's what that means. And backing up just a little bit, I want to point out that most every tool in David will point out what list you're currently working on, demo list 2, what background, because again, this is very important to know where we're at in our analysis. And then it'll give the list of, or the number of David IDs. Once we upload our list into David, everything else from here on out is done via David gene ID. So we can see kind of a discrepancy between these 390 Homo sapiens genes or identifiers. These are the numbers of Affymetrix IDs that I uploaded. However, those 390 get translated into 372 David IDs because certain ones of those identifiers actually map to the same David gene ID. So I just want to clarify that. That's important. So again, this percentage is based on the David gene ID. And then I've got a couple of other bars here, a chart bar and then a blue bar. So there are several annotations through here. Gene ontology is very popular, and I just want to say something really quick about that. There are several levels of gene ontology that David has in the knowledge base. These ones that are highlighted in red are defaults that we found, even in the other categories, that we found to be very useful in the analyses. So if you don't know a lot about these databases, it's a good way to move forward just using these defaults. And those can be checked and unchecked up here. I can clear them. I can clear my own selections. I can select whatever databases I want to use, but I'm going to stick with the defaults for this. And then back to the levels of Go, because this is a question we get often. what are these? So BP is biological process, CC cellular component, MF is molecular function. Direct means it's an annotation mapping to a gene that we got directly from one of the sources like NCBI or UniProt. Gene ontology is a hierarchy, or it's hierarchical. So given a term, we can relate that same gene mapping to all of its ancestor terms, its parent terms, its grandparent terms. And as we do that through the whole hierarchy, we now expand that annotation to this all category. And then because it's hierarchical, if we think of like a pyramid or a triangle, the top level, the tip of that pyramid would be level 1, 2, 3, 4, 5. Obviously, there's more than 5 levels, and all of that would be in all. But that brings me to this fat category. So several different groups have created what they call Go Slim. So they try to filter down the number of Go terms, but in the case of Go Slim, they're typically looking at the broad categories, the earliest ancestor. But we found that the more specific terms were more useful when we did our analysis. So we created this fat category, which would be more of the base of that pyramid into the middle of the pyramid. But by default, we check the direct. So I'm not going to go through all of these due to time, but I'll take questions at the end. Okay. So one of the first tools that we can look at that has no statistical analysis involved is this functional annotation table. So I'll open that, which opens in a new tab. And basically what this is takes every gene that has some kind of annotation based on my selections in terms of those categories, and shows me what annotation per database is associated with that given gene. So it's one gene at a time. I can see my Affymetrix ID, I can see the gene name, related genes again, the taxonomy, and then each of the categories with several terms that have been mapped to this specific gene from the databases, the original source databases. And these are linkable, so I could go out to QuickGo and get some more information about that. So it's a very simple way to quickly look at what genes I have and what kind of annotation are associated with them. However, with a list of almost 400 genes, that can be kind of tough to understand the biology behind it. And this is why we developed David, because looking at these big lists of genes, one gene at a time, and trying to figure out how they're associated with each other is difficult to do by hand. So one of the first functions in David for enrichment analysis was this chart function. So let me open that. So what this tool is doing is looking at all the genes that are associated with a given annotation term in my gene list, and then comparing that to all the genes in the background. In this case, it's Homo sapiens. More specifically, it should be U95A microarray, because what you want your background to be is all of the genes that you had some kind of chance of selecting in your experiment. So in the case of, some of these human genes, maybe they weren't on the array. So ideally, I would want to use the array as the background. But we find that, most experiments use almost genome-wide sets of genes. So it's not that big of an issue to use it here. But ideally, again, you want to use your background to be the group of genes that you could have selected your gene list from. So one way we can compare this is using a Fisher exact calculation. And that's what would be in this p-value column, which ranks this list here. However, this is a modified Fisher exact. And what do I mean by that? If we go to some of the help, there's a little example here. We found that in the case of, I'll call them one-hit wonders. So there is one gene associated with a given term. Sometimes those p-values can be very inflated, but the fact that we only have one gene on our list, it gives us pause, right? We don't trust that. So if we simply subtract one from the number of genes that are associated with a term, it takes care of that situation. So that's the modification. So this 2x2 contingency table uses 4 key pieces of information. It uses list hits, which is the number of genes in my list that is annotated to a specific term. It uses list total, which is the number of genes in my list that's annotated to the category. So for instance, go term BP direct. And then in the same way, population hits the number of genes in my population that map to the specific term in the category. And then population total, the number of genes that map to the category from my population. So it's very important that your gene list is a subset of your population and that your gene list is, or your population is the set of genes that you could have potentially selected in your experiment. Okay, so I won't go too much more into that. It'll take too long. Nonetheless, we get this ranking. So protein binding is very important, or very enriched, let me say, in my list of genes. And actually, we see several terms that are very similar: identical protein binding, protein domain specific binding. And I'll see these terms for all the categories that I selected. In this case, it was the default categories. Some other parts to this are related terms. So I'll go ahead and click on that. And this one is not very interesting. So let's pick a better one. Let's do cytokine receptor interaction. So I'll click on related terms. Okay, so now I get several terms, the first of which is the term itself. Now this is just like related genes, except we're flipping it. We're looking for related terms using this kappa statistic based on the genes in our gene list that are in common to it. So we can see the first two here, cytokine receptor interaction is the actual term I clicked on. The next one's viral protein interaction with cytokine and cytokine receptor. This one has an issue, UniProt keyword, but it's actually cytokine. I don't know if you can see that when I mouse over. Immune response, which is very related to that. So this gives us the related terms. And again, it's using that kappa statistic. And I'll come back to that because that's important moving forward. We also have this blue bar, which just represents the genes in my list that are associated with this. So if I click on that, I get kind of a gene report, kind of like the batch gene name viewer, but specifically for the term that I clicked on. Let me go back up to where we have a header. We have a count. This is just our list hits. We have percent. It's just a percentage of our list. And then I've talked about the p-value, which is the modified Fisher exact, and then a Benjamini, which is for multiple test correction. Now, if we look at options, we have several options here, including other statistics for multiple test correction. We can actually use take a look at the non-modified Fisher exact. We can get all the counts that go into the Fisher exact or the modified Fisher exact, and we can kind of control the number of records that we see. This thresholds piece is based on what results are returned. So in this case, I do not want to see any terms that do not have more than two or more genes from my list associated with it. The ease is actually this p-value, it's the modified Fisher exact. I want to see everything that's 0.1 or less. So I can play around with those. One other thing to point out here, I can create a sublist from these genes. So the genes that fall under this, or I can select several. But I'll select the first two, and then I can create a sublist. I can give it a name. Okay, and then if I go back to my list manager, I now have this list in my list manager that I can then use and do maybe some kind of drill down type of analyses with it. But I'm going to stick with demo list for now. So we've covered the table, we've covered the chart, which gives us our enriched biology. And actually going back to the chart, just to point out again, there's a lot of terms here, right? A lot of enrichment here, but several of these terms are very similar to each other. We saw that with the related terms. We can see it in the name, protein binding, identical protein binding. So this caused us some issues early on with really trying to figure out what our lists mean, what are the biological themes that are enriched, because we still have to go through all of this, and it can be difficult. So then we came up with functional annotation clustering. Okay, so what this does is it takes that chart report and it uses that kappa similarity score, and then we created a fuzzy clustering heuristic to use that similarity score to pull together similar terms. So I mentioned that this list was from GP120 in HIV, inducing chemokines, kinases, transcription factors. And the first cluster we see talks about transcription in several ways. So what this, I won't go into the clustering algorithm right now because it's a little too much, but again, this is using the kappa similarity score between terms and then trying to cluster those together. And the fuzzy part of that is that we know that genes can participate in several different biological processes and functions. So we wanted that to be reflected here, that the biology is really is fuzzy. So you may see the same annotation term in several different clusters here to reflect that. Other than this, it's similar to the chart report. So we have the category, we have the term, we can get to related terms, we can get to the genes that are mapped to this term, the count, the p-value, Benjamini, all those same things. But additionally, there's this enrichment score up at the top, and this is what these clusters are ranked by. So if we take all of the p-values here, the modified Fisher exact e-score, and we can do a geometric mean on those and create this enrichment score. So it's a way to try to rank the enrichment of the cluster itself. So that's what the enrichment score is. Some other things, this G button just points to all the genes that are associated with the terms in this cluster. This icon here is very big. Let me pick a different one. Let me pick this cytokine activity. So what this is showing me are the three terms that are in that cytokine cluster and how they are associated or not associated with the gene. So green here, if I mouse over it, I see the gene and the term name, means that there is a mapping between this gene and this term. It's annotated to this term. Black means that it is not annotated. So now we can kind of see the groups of genes and how they relate to each other. And again, that goes kind of back to the kappa similarity score. So it's kind of like a binary matrix for that. Okay, and just like with chart report, there are several options you can use here to control the kappa similarity, to control the clustering. I'm not going to go into this too much just because of time. Some of the display options we saw on the chart as well. We default to this medium stringency, which controls several of these parameters, and that's a good place to start, but you can play around with it to get different specificities to your clustering. And something else I haven't mentioned is that all these tools have a download link. So if you click on it, actually you'd probably want to right click and save as, But it's just a tab delimited file. Again, most every tool in David has this to where you could download the information that's shown in the web interface. All right, so that takes care of the annotation summary and the functional annotation tools. So now I'll move on to gene functional classification. So this is very similar to what I just showed with the functional annotation clustering. It's using the same fuzzy heuristic clustering algorithm. It's using the kappa similarity, but just like the difference between the related terms and the related genes, it flips it. So now I'm looking for groups of genes in my list that have very similar annotation. So they're participating in very similar biological processes and functions. So that's what I'm seeing here. Again, not surprisingly, my first cluster of genes are a bunch of transcription factors. I can see some chemokines here. There are some kinases here. So this is really, what my list is about. The demo list too. All the same similar types of options that you have here to control the similarity score, the classification. Now, one thing I didn't mention is that the functional annotation clustering uses the categories that you selected. So in this case, or in the case of what I just showed, it was the default categories. But in the case of the gene functional classification, because the clustering is so computationally intense, takes a lot of resources, we limit that, and it cannot be controlled by the user. And most of these are the default categories that I showed, but it's just a way that we're able to get the best clusters for the least amount of cost, basically. So very similar, again, to the functional annotation clustering, we have an enrichment score. And again, that is based on the terms that are behind this and the enrichment of those terms in my list. Related genes. So the difference here for this RG is we're treating this whole cluster as kind of one agglomerated gene. So any terms associated with any genes in here, the terms that were used to cluster these, are going to be used for this related genes function here. Terms are just the terms that brought these together. A few things that are different in this view. Of course, we have our category, our term, which links out related terms. So that's the same thing that we saw before. Genes in group, and then genes in not group. Genes in group are the genes in this cluster that are associated with this term. Genes not in group are the genes in my list that are associated with this term, but did not make it into this cluster of genes. And then I think everything else here is very similar to what I've already shown. Okay. And then just like with the term clustering, we have this 2x2 kind of table view where you can see the association between the genes and the terms. Okay. So next, actually, let me back up because there was one thing that I missed that's important. I'm going to go to functional annotation chart one more time. I'm going to show it as a tab. So I mentioned how these most of the annotation terms are linked out to some other information. In the case of KEGG pathway, Biocarta pathway, those actually have a pathway viewer within David. So those links go to a pathway viewer in David. And then recently, we also implemented wiki pathways. So let me go back to pathways. I'm going to clear these. I'm going to select KEGG and wiki pathways. Recently, we've added wiki pathways, and we've also recently created a new pathway viewer for wiki pathways. So I'm going to show that real quick because that's a very useful feature in David. Okay. So here I have my wiki pathways, KEGG pathways. It's a chart showing me the enriched pathways in my list. I'm going to look at cytokines. So if I start with KEGG, I see this cytokine, a cytokine receptor interaction pathway. It's a pretty big pathway. And a couple of things that we can see here are these blinking red stars. And what these denote are genes in my list and where they are in this pathway. If I scroll down, I get a list of genes that are in this pathway, and those that are in red are the genes in my list. So these are all the pathway genes, and then the red ones are ones found in my gene list. I can stop this from blinking here, and I could take a screenshot right now for this older style of pathway viewer in David. That's the best way to do it. If I want to zoom in and out, I'm going to use my browser to do that. There's also a link to KEGG. So now let's take a look at wiki pathways. So this is the newest version, the latest and greatest version. So again, this looks very different from what I just showed. A big part of that is the difference between wiki pathways and KEGG pathways. But another piece is this moving dashed line. Instead of those red stars, we're now highlighting the genes in your list by having this animated border around the gene in the pathway. And those are actually colored based on that value column that I included in my gene list upload. So now all those were 2, so it's like a fold change of 2. So that's still kind of in development, I'd say. But some other things that we can do with this, again, we can stop this animation or start it. We can zoom in and out. So we're not using the browser controls anymore. We can use it within the pathway viewer. Some of these pathways are very big, so I can drag it, pan it around, drag it around. I can reset that view. I have exports for different formats here. And then, of course, we have the link out to Wiki Pathways itself. Similarly, we have the genes in the pathway, and those shown in red to be the genes in my gene list. And you can search those, you can sort those, so on and so forth. So hopefully a little bit more user-friendly, and hopefully eventually we can go back to KEGG and maybe implement this style in KEGG. All right, so getting to one of the last tools here, gene ID conversion. So I mentioned in the upload, if you weren't sure about your ID type, you could go to this not sure identifier type, and it would take you to this this conversion tool. And the conversion tool is meant to convert between different ID types, but in the case of not sure, it can actually help you figure out what ID type you have. So I'm going to take my Affymetrix list that I've already got loaded, and I'm going to convert this to an Entrez gene ID. I need to put in a species, again, kind of like with a gene symbol upload, and then I'll submit that. Okay, this tool is not the most efficient. It can take a while, so be very patient if you use this, but I promise you eventually you'll get there. Let me close some of these tabs. Okay, eventually you'll get to something that looks like this. On the left-hand side, there is a summary of the success of the, or non-success of the conversion. So this first line is showing me that 390 of my Affymetrix IDs were found in the database and they could be converted to an Entrez gene. The second line would be if some Affymetrix IDs were found, but they couldn't be converted to the Entrez gene in this case. Or could I not find them in the knowledge base at all? And then the final category is ambiguous, which would mean that my ID looks like a couple of different ID types. And in that case, I would be presented with some information down here showing what potential types they are, and then I could choose to manually convert those here. But what we're presented with, since we had success for all 390 IDs, is our original Affymetrix ID, the Entree gene ID that it was converted to, the species, and then the David gene name. So again, I can download this file if I choose to. Again, tab delimited, open it in, oops, sorry, back here. Could open it in Excel or whatever. And then I have some other choices here. I could submit it to David as a gene list, or I could submit it to David as a background list. And this is kind of getting back to that not sure option where, you know, if I wasn't sure what my identifier was, I now was able to convert it and figure it out. So let's submit it back to David. So I'm going to go ahead and do that. I'll just take this default name, select OK. Then if I go back to the list manager and I scroll down, I can see this new converted list in here, which is actually the same list in this case. All right, so finally, there's a couple of tabs here. Since this is a coding group, from what I understand, maybe you would like to code using David. So there's two kind of programmatic ways, I guess, to interact with David, other than the web interface. There's the David API, and it's very simple. It's very old. It just uses GET calls and links to David to use several of the tools. So here I point out it's not for high throughput. It's not for large gene lists because it's a GET function. Your gene list will get cut off if it's too big because it won't fit in the URL character size limitation. Again, this is an older functionality. However, you can see here several examples of how we could load lists into David and get to certain tools. In this case, I'm showing a list upload of Entree gene IDs, and then I want to get to that functional annotation summary. So I can click on that, and here I'm at that summary. My current list is the list I loaded. I can rename that to BI list if I wanted, and then I can actually go through David and use all the other tools that are in David. So I've got examples of several other ones. Tell you what, let's do a chart report just real quick. That'll be the last one. So here, so my list is uploaded. It's only 4 IDs. I can see all the annotations that are associated with them here. Okay, so if we wanted to get a little more complex, we do offer, this is a little bit older too, it's stateful David Web Services using SOAP as the protocol. And so here's just kind of an overview. You could create using the client stubs, you could create your own code in whatever language you want that can interface with this SOAP. So Java, C, Perl, Python, R, MATLAB, so on and so forth. And then you can make calls to David, which will go through the web service and then use all the tools that I've talked about today, which of course interact with the David knowledge base, and then get some information back, and then you could parse that and use it however you would like. So this would be the way to use to upload larger gene lists, multiple gene lists, things like that. I've included a slide on how to get a chart report. So it goes through, you know, connecting to the client here. You have to register for this service. Actually, let me back up to that just to make sure everybody can see it. So in the David, under Technical Center, I'm sorry, downloads and APIs, there's David Web Services, and then there's this register here. Just some simple information. That's just so we can keep track of anybody that maybe is abusing the service and taking resources away from everybody else that's trying to use David. So again, I won't go through this now because we're running out of time, but if you have any questions on any of this, this is just a simple Python client code that uses the David Web Service to upload a gene list. look at certain categories, and then get that chart report. So actually, down in this bottom right corner should look similar to the chart report that I showed you. You can see the protein binding, identical protein binding, so on and so forth. All the same stuff that we got through the web interface. Okay. Weizong, did you want to take it from here? You want me to go through the last slide?
You can do it last slide.
Okay. All right. So David's been around for over 20 years, which is amazing for a bioinformatics tool. It actually, I think it predates the word bioinformatics maybe, but a lot of people over the years have been instrumental to getting David going, getting it out there, and continuing to support it and helping it live on. First of all is the users of David, and any feedback is very welcome. We know it needs some updating and things like that to make it a little bit more user-friendly and more up-to-date, but feedback from the users is what gets us there the quickest and best. But other than that, NIAID and NIH, Dr. Cliff Lane has been extremely supportive over all of these years. Dr. Basler with the Frederick National Laboratory, our lab head, Dr. Imamichi, and everybody in our group, the IT support. So this takes a lot of resources and updating and things like that. And this group under Sharon Beck is very instrumental in that. The group at the Advanced Biomedical Computational Science are some collaborators of ours and have been instrumental in David over all of these years. And then, of course, all of the past David team members that came before and helped develop David. Thank you. Okay, Alex, guess we can take questions.
Brad, that was an amazing demo, and I just wanted to thank both you and Weizong for being here and providing that introduction and demo for us. There are quite a few questions in the chat. I can kind of read these out to you if you'd prefer that, and then hopefully some others can maybe unmute themselves and ask questions as well.
Sure.
All right, so I think the first question here is, what is the main advantage of David compared to other methods? And they specifically mention a method called MAGIC.
MAGIC. I am not familiar with MAGIC. There are a lot of different tools and methods for looking at lists and understanding the enriched biology out there. And there are several great tools out there. We hope for David that it is very user-friendly. I think that's what's helped it survive over the years, is to be user-friendly. Now, I know, as I mentioned before, there's some updating that could stand to be done. But I think that's one advantage. Another advantage is the background knowledge base. So there are a lot of groups studying non-model organisms that can actually get annotation from David because we include so much in it, and we try to support everybody. So those, I think, would be two of the main things. But not to take away from any of those other tools out there. There are many great tools that do similar types of things, and I would encourage people to look at those simultaneously in their analyses, because there can be strengths in one and weaknesses, and then the same in others, and to look at those together, I think is very useful.
Great, thanks. The second question is the fold enrichment in the functional annotation and enrichment compared to background, sorry, is the fold enrichment in the functional annotation and enrichment compared to background? And they're referring to the functional annotation chart.
Okay, perfect. Yes. That's exactly what it is. So it's very simply the percentage of genes in my list annotated to a term divided by the number of genes in my list annotated to the category, So a pathway in KEGG versus KEGG itself, all pathways in KEGG, divided by the same ratio from the background. So it's an enrichment over the background. So if I have 10% kinases in my list, and in the population there's only 1% kinases, there's some kind of an enrichment there. So that's what that's supposed to be.
Perfect. There's a question about looking at genes in the context of a pathway chart. Pretty sure you already answered that. Not sure if that person wants to elaborate further on their question.
Yeah, that has already been answered. Thank you.
Okay, great. Thanks. And another question, should upregulated or downregulated genes be processed separately?
That's a great question. I don't know that I would be the best expert on that, but from my experience, it's good to look at it in all angles. So look at upregulated by itself, look at downregulated by itself, but then definitely look at them together. And what you can do, and David, I showed that combine function in the list manager. You could upload those two lists separately and then combine them within David. But I definitely would suggest looking at it from all angles, because certain pathways, it's important to see that certain genes are upregulated and certain genes are downregulated.
All right, thank you. And the last question in the chat, if I copy a gene list with a second column of values from Excel, will it recognize as tab delimited?
No. In the paste, it will recognize that second column as just one of the other identifiers. So if you want to do that, you need to upload the file. But from Excel, you could either copy it into a text editor and save it, or just save it in Excel as tab delimited.
All right, perfect. And that was the last question in the chat. Are there any further questions? Feel free to unmute yourself. You can also post those in the chat. And I'll just, as a reminder, that we are recording today's session, and that recording will be made available within 24 to 48 hours. So you can all refer back to the recording as well. All right, we have a new question in the chat. Would you recommend David or GCEA and Metascape? Oh, sorry. Would you recommend David over GCEA and Metascape?
No, which I guess I should, right? But there are different ways of kind of looking at the same thing. So again, I think there's strengths and weaknesses to both. I think there's going to be a lot of overlap between those. But I would encourage people to look at it from all angles. Again, use those different methods, see what's different, because again, you may see something in one that you don't see in the other.
All right, great. Any additional questions? And Brad, I think you touched on this. I just have a quick question.
Sure.
The David knowledge base, how often are you guys updating that?
Quarterly. And we're coming down to getting one out in the next few days, hopefully, to meet that. But yeah, we shoot for quarterly to update those.
Okay, great. That's awesome. All right. Well, I do not see any further questions in the chat. So if everyone could join me in thanking our speakers again for being here today. You can, unmute yourself, say thank you, or, give a round of applause. We really appreciate them taking the time to show us their tools. And thank you so much for all of our attendees for being here. So I am going to go ahead and close out the recording.